let me go to the board and explain to you that what happens in a DNA sequencing reaction. Okay, I will be so happy if you will tell me what is required in a DNA replication reaction. Okay, so I'm looking forward to hear from you. If we have an empty append of tube, so what should we add to that tube to initiate a DNA replication in vitro? So what are the components? Can you tell me? 1, 2, 3, 4.
Sir, nucleotide DNTPs or sir DNTPs
Primers.
tag polymerase होगा सर.
Anything else?
Template DNA.
Exactly. This is what I was looking for. Template. Okay, so these are the 5 major components of the DNA replication reaction in vitro. Okay, so if I wipe it away now, so I hope everybody knows these 5 component system for DNA replication. Okay,
सर इसमें टैग तो यूज नहीं होगा, टैग polymerase.
We have written here DNA polymerase at the moment. So
Okay, sir.
we will talk about tag polymerase on the 3rd generation DNA sequence. Okay, so when I write DNA polymerase, it means that polymerase is working at 37 degrees centigrade. Okay, so that is the 1st generation DNA sequencing, and that's very important to tell you rather than jumping to the next thing. Okay, so you will understand that how these things moved on, and this understanding is actually very, very important. So you have double-stranded DNA, you heat denature it to single strands, and then you have primer in this 5' to 3' direction, which will run on the template, which is from 3' to 5' here. And on this one, the primer is 5' to 3', and this is a 3' end of DNA, this is 5'. So you can easily see that DNA synthesis runs from 5' to 3' on the fresh strand of DNA. On the template, it runs from 3' to 5' direction. Okay, never get confused for that. It's very much visible here. It is very much visible here. Okay, now this is a normal polymerization reaction, and this is the new strand that you are getting, this one. Okay, this is now the new strand. This is normal DNA replication, and you should be very clear about DNA replication before we go into DNA sequencing. Okay, so let me ask you some questions here. I'm drawing a structure on the board, and I hope you can see it. So this is carbon position 1, carbon position 2, carbon position 3, carbon position 4, and then carbon position 5. Now, what are these molecules?
Sir, this is the ribose sugar.
Sir,
Sir,
OH.
this is the ribose sugar.
Are you sure this is ribose?
Sir, it has five No,
It is deoxyribose. If you have to see
because of the OH. Yeah.
if I add OH here, it becomes
Ribose.
ribose with OH. If I remove this OH, it becomes deoxyribose. Okay. And what does it become if I remove this oxygen from here as well?
Dideoxyribose.
Yes,
Dideoxy.
it becomes dideoxy. We know that DNA polymerization reaction occurs between the two nucleotides, and one of the nucleotide must have an OH group and the other nucleotide must have triphosphates at the carbon position number 5. And this triphosphate position will actually, on the other nucleotide, will bind at the OH position. Okay. If there is no OH here, there will be no binding between the 3', the carbon position 3, and the carbon position 5. The other thing you must know from this structure is this is the position where base is attached. Okay. And this is the position where the new nucleotide will attach. Okay. And this is alpha phosphate, this is beta phosphate, and this is gamma phosphate. The phosphate closest to the carbon position is called alpha. Okay, and this is very important in identifying the radioactive nucleotides which are selected for DNA sequencing purposes. We use alpha 32P. Okay, so the nucleotides which are labeled with 32P at alpha position. Okay. So that was the first generation sequencing where only the radioactive nucleotides were available and those were used in sequencing reactions. Okay, I hope you understand all this. And my main emphasis is that if you don't have an OH here, there will be no extension of the chain. That is, chain is terminated. Okay. Now I'm coming back to the sequencing reaction. Okay. We already know that it has got 5 components in it. 5 components. We have a single stranded DNA. The primer is bound here. Okay. And then in the 5 component system, one of the component is deoxy NTPs, DNTPs. And DNTPs, they are deoxy ATP, deoxy TTP, deoxy CTP, and deoxy GTP. Okay. These are the 4 different nucleotides. Now, what is done in a sequencing reaction is that we also have dideoxy ATP in there, dideoxy TTP in the reaction, dideoxy CTP, and then dideoxy GTP. A sequencing reaction, in addition to the deoxynucleotides, also has dideoxynucleotides. Okay, it means that either the deoxynucleotide can be added to the growing chain here, or either a deoxy or a dideoxynucleotide. Any one of them can be added here if there is a T on this strand. So this may be dideoxy ATP, which is being added here, right? Okay. So, or alternatively, it can be a deoxynucleotide. If there is a deoxynucleotide added here, the chain will keep on growing. But if there is a dideoxynucleotide here, the chain will not grow because it has a hydrogen group at carbon position number 3. And the hydroxyl group is not available for phosphodiester bond formation with carbon position number 5 at the coming nucleotide. Okay, so now just keep your mind open. What I'm going to ask you. I said in the growing chain, just after the primer, either a deoxy ATP may be added or either a dideoxy ATP will be added. If dideoxy ATP is added, chain will terminate. If deoxy ATP is added, chain will not terminate. So the second nucleotide will be added. Okay. Now, if you just imagine that this mixture of the deoxy and dideoxy have a ratio of 1:1 in the sequencing reaction mixture, what will happen?
प्रोसीड ही नहीं होगा. जो DNTPs हैं वो एक होगी तो वो आगे अगर मिसाल के तौर पर 2 DNTPs की जरूरत हुई तो वो कंज्यूम हो जाएगी और रिएक्शन रुक जाएगा.
What, in other words, you mean that there are 50% chances here, 50% chances that either a dideoxy will be added or a deoxy NTP will be added. Okay, so for example, the chain is growing like this. 50% chances say that at this position, this DNTP will be added. Okay, if the DNTP will be added, there are chances that once another nucleotide will come in, there are 50% chances again. Okay, so there are 50% chances here again 50% chances that either a deoxynucleotide can come in or dideoxynucleotide can come in. 50% chances. Okay, if this concentration is 1:1, what you are going to get? In this too, what is the probability again? If the reaction is not stopped here, not stopped here, it will definitely stop here. So earlier or at this position. So if we have equal concentration of both DNTPs, what we will get? We will get primer. This is straight line is primer, and the dot indicates addition of one nucleotide with the primer. Okay. The primer is this one, and then 2 indicate 2 nucleotides are added. The third one indicates 3 nucleotides were added. because there are equal concentrations of the two DNTPs, most of the end products, means the fragments which we will get, they will have only few nucleotides added to it when the chain has been terminated. So we get just short fragments. And we will read the sequence very close to the primer. When such fragments are generated with only few nucleotides addition, even 10 nucleotides or 15. So we are reading only the first 15 nucleotides which are closest to the primer sequence from here to onward. Short fragments are obtained if we have equal concentration. If we have double the concentration of DNTPs, so there are 66% chances that the chain will terminate here. If we reduce the amount of DNTPs, what we let the reaction go, how the reaction is controlled by the concentration of dideoxy in the sequencing reaction. Okay, so if you have this ratio, for example, 1 to 10, that there are 10 to 1. 10 to 1 means deoxynucleotides are 10 times in excess to the dideoxynucleotides. So there are these chances which were increasing. By increasing the dideoxynucleotides, the chances were from 50% to 60% or 66% here. By increasing the deoxynucleotides in a sequencing reaction, you can actually increase the fragment sizes. Okay? It means you can read more nucleotides. I told you that there is a technical problem that we cannot read first 25 to 50 nucleotide sequences close to the primer. We can't read that. It's due to this reason. That in order to get 1,000 nucleotide sequence, you have to decrease this dideoxy concentration in your DNTPs so that you should get longer DNA fragments. You should have a decreased probability, 1% chances that a dideoxy can be added, 99% chances that a deoxynucleotide will be added, a chain will grow more. Okay? I hope this makes a sense. That higher is the dideoxynucleotide, chain termination will stop earlier. And you can't read DNA sequence for a long DNA fragment. But if you reduce dideoxy, so the reason that whenever you use this mixture, what is labeled? Which one is radioactively labeled, fluorescently labeled, or a color dye labeled? It is this part. It's only the dideoxys which are actually having the label on them. Okay. Incorporation of deoxy ATP does not give any radioactivity to the growing chain. Only the terminator, chain terminators will tell you, okay, that this is the stop point at the chain length. Okay, let me say it in other words. when I make a drawing on this board, I take one molecule, one strand of DNA in actually millions of the strands of DNA which are present in a tube, millions of templates. So when we discuss about just one fragment. Okay. So out of a million DNA fragments, there are thousands of fragments which can be terminated at the same point. This is the primer. This is the first nucleotide, which is dideoxy. First nucleotide, which is dideoxy. First nucleotide, again dideoxy, and first nucleotide, again dideoxy. Out of the millions of molecules, single-stranded DNA, there are many, many DNA strands in the sequencing reaction which are terminating at the same point. Okay. And they are terminated by the dideoxynucleotide. Suppose your dideoxynucleotide is color labeled. Universally, what happens is a GTP is always labeled with a yellow dye. A CTP is always labeled with a blue dye. A TTP is always labeled with a red dye. And an ATP is always labeled with a green dye. This is a general rule all over the world. In the DNA sequencing, if you see a green peak, it means it is ATP. If red peak, it means it's a TTP. Okay. So if you are using the dideoxynucleotides as colored molecules, what will happen? If there is a T here and dideoxynucleotide, which is ATP, has been added here, in all the cases, it is the same. One, two, three, four fragments have terminated with dideoxy ATP, and dideoxy ATP is a labeled compound which has a color, and that color is green. So each of the fragments which is terminator in millions of the fragments, they will end up with dideoxy ATP. Okay, the length of the fragment is primer plus one nucleotide. Is it all right? So what I will write here is green G. Okay. Now there are many other fragments which will not get dideoxy ATP, so the chain elongation will continue. Okay. Chain elongation will continue to the second nucleotide. Now, you knew that there is a T here because this is identical DNA strands. So all have T at that position. Suppose there is a G after T on the template strand. What will be added here? C will be added here, right? And if this is a dideoxy CTP, what is the color of C? CTP is blue. Okay. Because here there was no dideoxy nucleotide addition at the first position, so that chain is not having any color for A. The color will come only when a dideoxy will be added. Okay. So dideoxy CTP addition is going to give a blue color. to the chain. So it means that the second fragment that we obtain in a DNA sequencing reaction is having this nucleotide plus another nucleotide. This fragment is ending at green color, and this fragment is ending at blue color. If the chain elongation proceeds 1, 2, 3, and suppose this third one is C is here, G is here. The third one is addition of G, which is yellow in color. Okay, so I should rather do like, yes, yellow color. So this is yellow. So you are getting nested sets of DNA fragments. What does it mean? It means primer plus 1 nucleotide, primer plus 2 nucleotide, primer plus 3 nucleotide. Okay, and this is now a yellow fragment. These are nested sets of DNA fragments which differ from each other by 1 nucleotide. So if you run these nested fragments ended with different colors, okay, on a gel, suppose this is the gel. Suppose we have a gel here. And we have these 3 fragments, primer plus 1 nucleotide, primer plus 2 nucleotide, a primer plus 3 nucleotide. Which of these DNA fragments will move fast?
The first one.
First one means green one. Okay, and followed by the blue one and followed by the yellow one. Okay. Now, if we can see the colors on the gel, what we will drive? This is a green, this is a blue, this is a yellow. And remember, sequence is always read from 5' to 3' direction on a slab gel. This is slab gel. So we had 4 different tubes. In one of the tube, we had DNTPs, all the DNTPs, and only 1 dideoxy ATP if it was radioactively labeled. If we have 4 colors, okay, we can do a single tube reaction. So in this tube, we have a single tube reaction, and we can run them on a single well. This is 60 centimeter long polyacrylamide gel, which is run at about 12,000 volts. That is a slab gel. The fragment which is farthest from the well is the smallest one, followed by the ones which are following. And each of these fragments, they are different from each other just by 1 nucleotide. This suppose if 20 nucleotide long is our primer. So this fragment is having 20 plus 1, 21 nucleotides. This fragment, which is gone to 2 additions, okay, it is having 22 nucleotides. The fragment behind that is having 23 nucleotides. So every fragment is different by 1 nucleotide. Okay, so these are the nested sets, and so on. It will go like this: 24, 25, 26, 27, 28 nucleotide long. Longer DNA fragment, shortest DNA fragment. Okay, and when we read the sequence, we read from bottom to the top. And if this is green in color, we will say the nucleotide sequence is G, this green represents ATP. Okay, so it is A, blue color is C, yellow is G, and so on. We will write on the sequence, and this will be the sequence of the newly sequenced strand. This one is the template, and this is the newly synthesized strand. Do you understand what I mean? Are you able to pick
Yes,
that
sir.
point? Huh?
Yes, sir.
Okay,
Yes, sir.
that's good.